Let us start with a new topic, that is reproduction in flowering plants. Reproduction in flowering plants. In flowering plants, mainly a sexual reproduction, and a flower is the most fascinating structure in the plant and is a reproductive shoot, mainly meant for sexual reproduction. Flower, which is a reproductive shoot, mainly meant for sexual reproduction. And a flower is having the parts, even the base of flower, which we have studied in first year itself. Till then, a quick glance over the parts of flower. As far as the definition of flower is concerned, is a reproductive shoot mainly meant for sexual reproduction. And let us see the parts of flower. First one, pedicel. This is pedicel. A stalk of flower is said to be pedicel. If the flower is with pedicel, they are pedicellate flowers. If no pedicel, they are sessile or non-pedicellate. Most of the flowers do have pedicel. They are all pedicellate flowers. If pedicel is absent, then they are said to be sessile flowers or non-pedicellate flowers. It is a stalk of flower. And the next part is thalamus. Thalamus. Thalamus is a floral axis on which the four whorls are placed. It's a floral axis on which the four whorls are present. That is thalamus. It's a floral axis on which all the floral parts are present. And in the flower, we do see four whorls of the flower. Whorls of the flower. There are four whorls. Two of them are accessory whorls, and two of them are essential whorls. Accessory whorls. One, calyx. B, corolla. And essential whorls of the flower. C, endocm. D, gynoecium. Totally four whorls. Those are calyx, corolla, endocm and gynoecium. Among these four whorls, two of them are named as accessory whorls. Why calyx and corolla are said to be accessory whorls? They are not involved directly in reproduction. Their works are either protection, even photosynthesis, even attraction. They are not involved in reproduction. That's why they are said to be accessory ones. And endocm and gynoecium, they are essential ones. Why they are said to be essential ones? They are said to be essential ones because they are directly involved in reproduction. Endocm is a male reproductive organ which produces male gametes. Gynoecium is a female reproductive organ which produces egg. So they are directly involved in reproduction. That's why they are essential ones. In which the calyx is a accessory whorl and is a first whorl of the flower, and this calyx is having individual units named sepals. Individual units named sepals. And these sepals may be free or fused. Are free or fused. If they are free, the condition is said to be polysepalous. If the sepals are fused, then the condition is said to be gamosepalous. If they are free, polysepalous. If they are fused, gamosepalous. And individual units of calyx are sepals. And usually the calyx is green in color and mainly meant for photosynthesis and protection. Photosynthesis and protection. That's all. And the next accessory whorl is corolla. Corolla is a most conspicuous part, the most attractive and the most conspicuous part in the flower. And this corolla is having individual units. Those individual units are named as petals. They are said to be petals. Even these petals may be free or fused. If these petals are free, polypetalous. If they are fused, gamopetalous. If they are free, polypetalous. If they are fused, gamopetalous. And these are petals. They are most conspicuous and attractive part in the flower. And they are variously colored. And their main function is protection and attraction. They are meant for the attraction of insects for pollination. The main function is attraction. Even another function is protection of inner essential whorls of the flower. In some flowers, calyx and corolla are undistinguished. They are undistinguished. If calyx and corolla are undistinguished, there is no distinction as calyx and corolla. Under that condition, they are said to be perianth. What is perianth? Undistinguished calyx and corolla is said to be perianth. In some flowers, we can expect undistinguished calyx and corolla. That is said to be perianth. Once the perianth is there, we are not going to mention its units as sepals or petals, but we mention those units of perianth are tepals. units of period are tepals. And here also, tepals may be free or fused. They may be free or fused. If the tepals which are free, that is, the condition is said to be polyphilous. If the tepals are free, the condition is said to be polyphilous. If they are fused, gamophilous. If they are fused, the condition is said to be gamophilous. If they are free, polyphilous. It's about accessory worlds. Calyx and corolla, their individual units, if they are free, if they are fused, the terminologies. And undistinguished calyx and corolla is said to be perianth. Its units are named as tepals, and there also, if the tepals are free, polyphilous, fused, gamophilous. And now let us go to essential whorls of the flower. Calyx is the first whorl of the flower, corolla is the second whorl of the flower, endocm is the third whorl of the flower, and gynoecium is fourth and innermost whorl of the flower. Fourth and innermost whorl of the flower. Endocm is the third whorl of the flower and is an essential whorl and is a male reproductive organ. This endocm is a male reproductive organ and its units are named as stamens. Stamens. These stamens may be free or fused. These stamens, they are the units of endocm. It's a male reproductive organ. And these stamens may be free or fused. If they are free, they are said to be free stamens. If they are fused, that is adelphous condition. Adelphous. If they are fused, that is adelphous. And each stamen is having the distinguished parts. At the apex, more or less a bilobed structure named anther, and the stalk, a stalk is filament. Anther and filament. In the anther, there is a production of pollen grains, and pollen grains are the carriers of male gametes. They are the carriers of male gametes. And now let us see about gynoecium. And a gynoecium is a fourth whorl of the flower and is an innermost whorl of the flower, is an essential whorl of the flower and is female reproductive organ. It is a female reproductive organ and its units are named as carpels. Its units are named as carpels, and carpels may be free or fused. If they are free, apocarpous condition. If they are fused, syncarpous condition. If they are free, apocarpous condition. If they are fused, syncarpous condition. And the number of stamens or carpels, that is varied. Stamens, they are varied in number, varied in size, and carpels, they are also varied in number and shape and size as well. And here the structure of carpel which we see here is, this is the carpel. The basal part is named as ori, and this is style, and upper portion is named as stigma. Stigma is a part which receives pollen grains, and they pass through the style and go to the ori. In the ori we do find ovids. In the ovids a egg, female gamete is there, where the fertilization takes place. This is a brief account of a vertical section of flower. With respect to reproduction in flowering plants, our target is to study about the structure of that stamen, even a TS of anther, where we could see where actually pollen grains are formed. That we have to see. Let us discuss about stamen. Already we know endocm is a male reproductive organ. It is having individual units named stamens. These stamens are varied in number and varied in shape and size. And these stamens may be free or fused. If they are free stamens, if they are fused, that is analogous condition. As far as structure of stamen is concerned, it is having two distinct parts, anther and a filament. Anther is bilobed structure, and each lobe is having two theca, and that is said to be dithecous condition. Ditheca. Anther usually bilobe, and each lobe is having two theca, dithecous condition. And anther is the structure where pollen grains will be formed. As two lobes, and each lobe is having two theca, so that it looks like tetragonal in structure. Tetragonal in structure, and there would be the presence of 4 microspores. To study about the internal structure of that anther, it is necessary to take a transverse section. Well, if we take the transverse section, we will see the structure like this. We will see the structure like this. And here we could notice 4 microsporangia. 4 microsporangia. And this anther is having 4 layers of cells. Those 4 layers of cells are outermost epidermis, next layer is endothelium, third 2 to 3 layered mid layers, and innermost tapetum. There are 4 microsporangium, singular microsporangium, and plural form microsporangia. This is one microsporangium, and each microsporangium is covered by 4 layers. Those are outermost epidermis. Next to epidermis is endothecium, and 2 to 3 layered middle layers, and innermost layer is tapetum. Among these 4 layers, epidermis, endothecium and middle layers, they are meant for protection of microspores and also help for dehiscence of microspores. Microspores are nothing but pollen grains. For the dehiscence, release of those microspores is under the control of epidermis, endothecium and mid layer. And the innermost layer, that is tapetum, it is a nourishing layer. It gives nourishment to the pollen grains or microspores. This is the innermost layer as well. It is mainly meant for nourishment. And then each microsporangium in young condition is having a homogeneous tissue. That homogeneous tissue is named as sporogenous tissue. Sporogenous tissue. Totally, that microsporangium is backed with a sporogenous tissue. Later on, each and every cell, they are all diploid, are capable to undergo meiosis, so that they get separated and they become in second stage. In the same anther, tears of anther, I have shown here the 4 stages of development of microsporangia. In the second stage, all the sporogenous tissue are capable to undergo division and they get separated. Now these are said to be microspore mother cells. They are diploid in number. Sporogenous tissue, diploid in number. We have seen these microspore mother cells. And these microspore mother cells, they are diploid in number. They undergo meiosis, leading to the formation of 4 haploid microspores. 4 haploid microspores, but they are present being attached. Hence, they are named as tetrad of microspores. Tetrad of microspores. Each microspore mother cell, which is diploid in number, which is diploid cell, undergoes meiosis, leading to the formation of 4 haploid microspores. And these 4 microspores, haploid microspores, they are held together, forming a tetrad of microspores. All of them are haploid in nature. All of them are haploid in nature, and gradually, in final stage, those microspores get separated and all the microspores are free now. And microspores are nothing but pollen grains. And these are haploid in nature. And at the starting we used to say microsporangia. But these microsporangia, when they produce the microspores, the same microsporangium is said to be pollen sac. Pollen sac. Once microspores or pollen grains are completely formed with the help of epidermis, endothecium and mid layers, a breaking of microsporangia takes place. Hence, the release of pollen grains outside. And these pollen grains are haploid in nature, but these are not male gametes. Pollen grains can produce male gametes. One pollen grain can produce two male gametes. Hence, that pollen grain is said to be male gametophyte. Male gametophyte. Let us discuss about pollen grain. Already we know pollen grains which are produced in microsporangia of anther. And pollen grains, they are haploid in nature and they are the carriers of male gametes. Pollen grains, they are produced in anther. Whenever we touch the flower, there will be a dusting on the fingers. As far as the structure of pollen grains is concerned, pollen grains are having a different architecture, shape, size, even design in different species. All the pollen grains in flowering plants are not unique. They do have different architecture, shape, size, even design. If you analyze the structure, pollen grain, their size is ranging around 25 to 50 micrometer. 25 to 50 micrometer. And pollen grains, they are covered by two walls. Those are outer exine and inner intine. Outer thick exine and inner intine. This exine is thick and is made up of sporopollenin, a very resistant organic material. It is resistant to high temperature, even the action of acids and alkalis. It can withstand the action of high temperature, even acids and bases. That's why it is the most resistant organic material, sporopollenin. The exine is made up of sporopollenin. And this exine is having the outgrowths, a rough outgrowths on the surface. And here and there, some minute pores are there in exine. Those are named as germopores. Germopores. Because when this pollen grain falls on the stigma of carpel, the inner content will coming out and become a pollen tube. That is through this germopores. Germopores. It is about exine. It's very hard, thick, made up of sporopollenin and is resistant to the action of temperature, acids and bases. And here and there we do find minute openings named germopores. And next to exine there is this one, a blue line, that is intine. And this intine is made up of cellulose and pectin. Cellulose and a pectin. And the innermost layer is plasma membrane. Plasma membrane. This plasma membrane encloses the cytoplasm, which encloses the cytoplasm. In this pollen grain, when it is just formed, is single celled. When it gets matured, we do see two unequal cells. The larger cell is named as vegetative cell, and a smaller, a spindle-shaped cell is named as generative cell. Generative cell. A larger cell is named as vegetative cell, and it is having a dense cytoplasm, even a reserved food, and a prominent, very large, irregularly shaped nucleus. Irregularly shaped nucleus. And then, in this vegetative large cell, a small spindle-shaped generative cell is floating. It is floating in a vegetative cell. It is also having a nucleus with cytoplasm, dense cytoplasm. But in vegetative cell, a large reserved food is there. And in 60% of plant species, pollen grains are shed or released in a two-celled stage. When they are released, they do have two cells. One vegetative cell, another generative cell. But in remaining 40% of species, the pollen grain is shed or discharged in a three-celled stage. In that 40% of species, this generative cell gives rise to two male gametes. Two male gametes. So that the pollen grain becomes three-celled. That is in 40% of plant species. But in 60% of species, just two-celled stage pollen grains are shed or discharged from the anther. This is about the structure. And as far as the viability, its lifespan, it differs from species to species. In case of cereals, pollen grains can remain alive around 30 minutes of their discharge. They will be survive up to 30 minutes in cereals like rice, wheat, jawar. But in case of the members of Rosaceae, Leguminaceae and Solanaceae, they can survive for a month. They will remain alive for about 1 month as well. But in case of cereals, their lifespan will be just 30 minutes. Within 30 minutes, they have to follow the stigma, and a pollen tube has to form, and a male gametes has to enter into ovule. That is about their durability. Let me have a quick glance over the structure. Pollen grain is a microspore or a male gametophyte. Their size is ranging between 25 to 50 micrometer, and they do have different architecture, shape, size and design in a different species. They do have two wall coverings. Outer one is named as exine, inner one is named as intine. Exine is made up of sporopollenin, and it is very hard and a highly resistant organic material. It is resistant, or it can withstand against high temperature, even the action of strong acids and bases. But here and there, some minute pores are there in exine. They are named as germopores. And next to exine is an intine. That intine is made up of cellulose and pectin. Is a thin layer. And next to intine there is a plasma membrane. That plasma membrane encloses cytoplasm. When pollen grains are immature, they are single celled. When they get mature, we do find two cells, and a large cell is named as vegetative cell. It is having maximum reservoir food and a large irregularly shaped nucleus, whereas a small cell is a generative cell. It is floating in a vegetative cell. It is spindle shaped, having a dense cytoplasm and a prominent nucleus. In 60% of plant species, pollen grains are shed or discharged in a two-celled stage. But in remaining 40% of plant species, they are released in a three-celled stage. How? The generative cell, it gives rise to two male gametes. So that two male gametes and one vegetative cell, three-celled stage. In that stage, in 40% of species, pollen grains are shed or discharged. And the durability of these pollen grains is concerned, it varies from species to species. In case of cereals like wheat, rice and jawara, they can survive up to 30 minutes. But in case of the members of Rosaceae, Leguminaceae and Solanaceae, the pollen grains can survive for a month. And even these pollen grains can be stored for a long time as a fossil because of that resistant organic material, sporopollenin. Even nowadays, just like blood bank, sperm bank, seed bank, even we do have pollen banks in which in pollen bank, pollen grains of some noble species can be stored. They can be stored in liquid nitrogen, where the temperature is minus 196 degrees Celsius. It is extremely very low temperature. In this liquid nitrogen, if the pollen grains are stored, they remain alive for a long time. Just like sperms, they are stored in sperm bank in liquid nitrogen only. And even seeds are also stored. In the similar way, even pollen banks are also there. In addition to this, some pollen grains can create some respiratory disorders like asthma, and bronchitis. If we inhale the flowers, pollen grains, in case they enter into our respiratory system, they may cause asthma as well as bronchitis. Bronchitis means inflammation of bronchioles, primary, secondary and tertiary bronchioles present in the lungs. They get inflamed. And asthma and bronchitis are very common. Even we know about one weed, Parthenium. In case we smell the flowers of Parthenium or the pollen grains of Parthenium, they enter into our respiratory system, that leads to pollen allergy. Pollen allergy. This is some sort of problems due to pollen grains. And one more interesting thing about pollen grains is that nowadays, as pollen grains are rich with nutrients, in Western countries, these pollen grains are made used as a tablets and syrups and used as they are rich in nutrients. Even tablets are available in Western countries. Syrups, in the form of syrups also they can take, and they believe if they take tablets and syrups, there would be high performance by the sportsman. Even these tablets and syrups can be used by the race horses, so that their performance will be very maximum due to the pollen tablets and syrups.